this Damir was making an intervention on this specific topic during this meeting with Emily, and we thought it would be really interesting to have him here. So Damir is a bioinformatician by training, and he has been involved for quite some time in some of the jewels of bioinformatics, JASPAR, which many of you know about, which is a collection of transcription factor and their specificity, and it has become a very important resource of bioinformatics, especially now that deep learning is making it possible to integrate increasingly large and complex amounts of information. Now, sorry, I have a little bit of background noise. And of course, unsurprisingly, Damir has been interested in understanding regulation, and that is going to be the topic of his talk today on a model system on zebrafish, which is obviously the kind of system on which you have everything you need, plus an extraordinary genetics that Damir is going to tell us about. And so last time we saw him, Damir was still a postdoc at Imperial, I think, but in the meantime, he has grown up and has become a PI in, I'm afraid in Croatia, but I'm afraid I'll be terrible at pronouncing the name of your institution, and I would rather you do it. But we are very, very happy to host you here today, Damir. And so the talk will be recorded. There will be a question and answer session after, but you may also receive other offline questions. And of course, I encourage all of you to join the NF Core Slack and to ask questions to our speaker and to interact via the Slack. And without further ado, Damir, the floor is yours. Thank you.
Thank you so much for the introduction, and thank you everyone for joining. It's my pleasure to be able to speak here. I will then start here just by sharing my presentation. And okay, are we seeing is everything fine? Can you just someone confirm?
Yes, it's okay. Yeah.
Okay. So thank you, Cedric and Jose, for actually for the invitation and for the chance to present this. So as Cedric mentioned, mostly I will be speaking today about the Daniel Code initiative, like what we achieved in the annotation of zebrafish genomics resources, and what are our plans for the future, what is our current status, and what we plan to do in the following years. And also, as I mentioned, like a little bit of since I recently started my group in at the Ruđer Bošković Institute in Zagreb, just a little bit of what my group is focused on and what we are doing with our collaborators, like from various institutes around Europe. So as you might know, until recently, there was like quite a couple of efforts to annotate the genomes of the regulatory landscape of model organisms, primarily ENCODE and human, and mode ENCODE for other, like Drosophila and C. elegans, and mouse ENCODE for mouse. And those efforts resulted with atlases of regulatory elements and functional annotation of these model genomes. However, there was no effort whatsoever to try to accumulate all the genomic resources available for zebrafish and to annotate the zebrafish genome, although the zebrafish is the third most complete vertebrate genome, and it's like an increasingly popular model organism used for over 1,000 labs worldwide in various specific to answer various specific biological questions, some of them which I will show you during my talk. So to circumvent this, like even before my era, my previous boss, Boris Lenhard and Ferenc Müller from University of Birmingham as leads. They formed this Zencode ITN, which one of the member was Matthias, who is also here with us. I'm happy to that I could have seen him today, because we work a lot on Daniel Code together, and like the foundation of the entire thing is basically Matthias, who is the guy behind it. So he is without him, like none of this would be possible. But also, like, it gathered like 15 other PhD students all across the Europe who work on this same topic, actually, to collect this data set and to make this atlas. And at some point I joined them as a postdoc at Imperial College and like started working on this. So with this effort, we gathered many genomics data. Some of them were raw data, which we annotate with the standardized vocabulary in the standard way, assign the metadata. We process them to map to the two recent versions of the genome, like including version 11, which came out during the process of the annotation using standardized pipeline and standardized way. And we made all these data and all this process data available to visualize, to download from the Daniel Code Data Coordination Center, but also like to visualize them in a UCSC and Ensemble browser. So, and this represents like the current state of the DCC, which we intend to expand in the future. So, as I mentioned, the DCC was developed primarily by Matthias, and without him it wouldn't be possible. And this is just the highlights how many samples we gathered and collected. We identified precise promoters, alternative promoters, and over 140,000 cis-regulatory developmental elements. So this is just one slide to show like all the people and who were somehow involved in all this effort, and who participated in this, with like people contributing the most being like in red. And this effort, and in the background there was is the photo from the final conference of the Zencode ITN doctoral network in Sunio, like, which was close by. We were finishing the analysis for this, and all this resulted in a major publication in Nature Genetics, where we actually presented the Daniel Code DCC, which link you can find on top here, and where we presented also the atlas of cis-regulatory elements of development in zebrafish. So just to say a quick words about that atlas itself and how it was developed. So as you might know, like the chromatin, the genome is characterized by some chromatin marks. So we have a taxic signal which marks open chromatin regions, or we have different chromatin marks which are typically found either on active promoters or on active promoters and non-active enhancers. But we also have some repressive marks which mark the transcriptionally silent chromatin, and it's not always easy to find which part belongs where, but fortunately there are algorithms such as ChromeHMM which give you this chromatin state and which can segment the genome based on the presence or the absence specific chromatin mark, and based on this combination of marks, it can be the entire genome can be functionally annotated. For example, if it has chromatin marks for active promoters or active enhancers, or if it's repressed or it doesn't have any mark at all. However, this approach has some slight drawbacks. So the resolution is like 200 base pair. The boundaries are not very well established. So to circumvent that, we use the open chromatin in a toxic defined regulatory region, and we function. But however, with a toxic, you don't get like the what function the region has. You only get the information about if it's open and active or not. So based on the chrome-HMM annotation, we gave this attack-seq region specific function, and we functionally annotate this. And here is like one example of the raw signals which we use on top of the chrome-HMM annotation, and like attack specific defined regions. So just a little bit of methodology. We defined these padres or predicted attack supported developmental regulatory elements as robust attack-seq peaks, meaning that they must be present, must be reproducible between two replicates. And we have two sets of them, one which is like developmental stage specific, and we have a consensus set to follow the dynamics and for comparative analysis between stages. And in this consensus set, we also had two different, one permissive, which included all the robust peaks, and one more strict, which required that they are open like in at least two consecutive developmental stages. And from this strict set, we got to like 140,000 developmental elements. So just to show you like a little bit of a picture, like summarizing what I all just said. So for each different stage, we have like this chrome-HMM annotation, and then we get the signal peak. We define the robust region, and then to each region we assign the function based on the chrome-HMM annotation. So in the end, like we came up with this way to visualize all this regulatory element based on their chromatin profile and the presence and absence of certain signal around the summit, and we managed to split and to isolate active promoters from active enhancers. So on this UMAP plot, each dots represent one regulatory element, one active region, and we managed to and they are colored based on the chrome-HMM annotation. And you can appreciate that we managed to separate active promoters to be fair, on two classes, because the model, of course, doesn't take the orientation into account of the promoter into account. And in zebrafish, the chromatin mark on genes are very, very biased toward the gene body. So you can find active marks only on the gene body, and like the upstream is quite unmarked with chromatin marks, unlike for example, mammalians. We find active enhancers. We also find primed enhancers. So these are enhancers which miss the active K27 acetyl mark, but they are poised for activity, like with K4 monomethyl. And we also find a lot of unmarked open chromatin, which either function as something else, which we didn't observe with our limited, unfortunately, amount of chromatin marks, or it is just poised for activation in some other stage. And for the consensus classes, we basically clustered all these 400 elements into different stages. So we find early, very early stages, very late, but also like a class of very constitutive elements which are open throughout the development. And using this approach of visualization, as I showed you before, you can appreciate that these early classes are open in the active enhancer region early on, and how the development progresses. They go to this inactive, like unmarked stage. The consecutive remain open throughout and marked throughout the development, and the late one, as the development progresses, they also like shift from this right-hand side, which marks the inactive or unmarked elements, to the more to this left-hand side, which marks the active elements. Also, we compared the evolutionary constraint of the elements. What we found out actually that the late one are evolutionary more enhancers are evolutionary more conserved compared to the early enhancers, which goes along with the hourglass theory of evolution. And not only that, but the enhancers are much more conserved evolutionary compared to the promoters. But the most conserved are the regions which are marked with repressive histomodification. So with the polycomb repressive mark, which we know that targets developmental genes important in the development. So we did a lot more of analysis, and I don't have time to talk about all of that, unfortunately. So for the Daniel Code paper and the genetics, Nature Genetics paper, I would stop here and like more focus now on what we are doing now. While we were preparing that paper, actually another paper came out with which says that they map cis regulatory elements in adult zebrafish, and they collected like different tissues and different marks from different tissues. However, their approach wasn't really like in this standardized way to make like the usable atlas that could be compared with our regulatory annotations. So Changhong Wan, PhD student from Ferenc Miller lab, who I'm helping in supervising, she actually took all these data sets and she reprocessed all of them using our standardized pipeline, which we used in pipelines which we used in Daniel Code, and now she did the regulatory block of this adult tissues, which we are comparing to our developmental regulatory block. And so she did for all of their tissues which she have, and assigned the function on all of this chrome-HMN state, and defined the attack regions. And these they are not padres anymore because they are not developmental, so we are calling pares, which is not as funny as the padre one, but still we have to call them somehow. And then now we have actually a comparable set to look with the development. When comparing with the developmental atlas, we noticed that most of the reused elements in adults are coming from brains, so a very vast majority of them, which is kind of expected because neurogenesis is the one thing in development which like is most prominent when you take the whole embryo as a whole. But we have also other tissues which show tissue specific activity. And a surprising thing that almost 4,000 elements are reused from development in all of the adult tissues. And when we look at their annotation state, like from embryonic development to the adult tissues, we see that mostly they keep their functional annotation. So the promoter will stay promoter, the enhancer will stay enhancers. However, we were actually quite surprised about this large proportion of enhancer which turns out to end up as promoter in adult tissue. And we were surprised, like, why would that be if it's biological or if it's an artifact factor like misannotation or something. So to answer that question, a student from my group, Marta Bošnjaković, she isolated RNA from 7 different tissues in zebrafish, and she did cap analysis of gene expression on this to get like promoters in all these tissues. She got like at least 15,000 promoters in each tissues, and which they were very well annotate, and they map into the annotated promoter tissues, and they separate well on the PCA as well. So we made like a very high confidence set of promoters, which can be used actually to validate the regulatory elements in the adult zebrafish. And what we find out actually that most of the promoters which were assigned defined by the chromatin overlap annotated assemble peak, and they are also supported by cage. However, there is a part of TSSs, like 4,000 TSSs, which are not supported either by assemble or by cage. And when we look where the annotation is, we see that most of the cage verified promoters are actually annotated in the ensemble promoter region. However, if they are not in the annotated promoter, they are mostly in the introns or distal. If they are not validated by cage, they are mostly in introns or distal intergening, which could indicate that those are actually misannotated promoters, enhancers, although a lot of them are in the first intron, which means that it might be that due to residual signal of K4 trimethyl, we have actually misannotation of enhancers in the as promoters. So we are, this is still all unpublished data, and we are actually currently preparing a paper on this. But in the meantime, we also wanted to explore different biological questions. So because zebrafish is also like a popular model organism for tissue regeneration, because it can very efficiently regenerate many organs like heart or brain. we wanted to explore these regeneration properties on a molecular molecular level, and include those annotation of these regenerative processes to the DANU code. So we established another doctoral network, which is called the DANU Recode. Like once, because we are restarting DANU code from scratch, and the other part because we are exploring the regeneration, and we actually got funded by Marie Curie Action. We established like several labs all over Europe who will work on this specific, who will find PhD students who will work on these topics. And we have a very well balanced group of mostly dry lab people and mostly wet lab people. And we want to explore also the possibility of comparing those molecular mechanisms and regeneration, genomic regulation in other systems which can regenerate, like axolotl, and also with organisms which are more relevant for human health, but they cannot regenerate efficiently as human cannot. So we are comparing to mammalian systems, namely with mouse, to try and that's where if there is a molecular mechanism which could explain like why mammals are not as efficient in regeneration like fish or amphibians. So with that, I would stop with the DANU code part, with just like what are the future plans with this. So we want to expand the DANU code data sets with new types of data, namely like with the explosion of the single cell data set nowadays. We not that we want to include it, only that we want to include, but we need to include it actually, because that's where the direction is going. We also like the pipelines we used are not really so much easily scalable or reusable like on the new data set. So I want to mention that we want actually to reanalyze all these data sets, like using NF core pipelines just to make this more scalable and more standardized, let's say, in a way, and easier for other people to reanalyze it and reuse it and to do all this analysis again. And also what we are exploring as a possibility actually to include DANU code and DANU recode in to the Funk data portal. We had like some good conversations with EBI regarding that, and with Emily as well, and hopefully we'll manage to include that, so that it will be under one big umbrella of animal genomics resources, and like with domestic animal, which would, I guess, be interest for this of an interest for this group as well. So I would just quickly, for a couple of minutes, say what my group is up for. So up to, so in all this thing, I explored a lot of chromatin data, but mostly we are interested in currently, we are mostly interested in core promoters and their elements and their function, and how different types of promoters, for example, sharp promoters, which are characterized by Tata box or broad promoters, which are characterized by CPGL and that can start from various regions, from various positions in the core promoter region, or developmental promoters which are responsive to long-range regulation. What are they sequence characteristic, and how can we define them? So this is what my group is currently exploring, and one of my projects called CRO-Age looks exactly into that, and it tries to solve the promoterome challenge by exploring core promoter elements in different vertebrate systems. So basically from Amphioxus, so like close relative of vertebrates from fishes to all the way to the mammalians, and to try to establish some kind of evolutionary rules which shape promoter function, and see how cancer or other diseases which are linked with the regulation, like revert these rules and like to end up in disease and disorders. So as a main essay to do that, we are using cap analysis of gene expression, which captures the using by biomethylating the 5' cap of the mRNA sequence and pulling it down. It captures like precisely the transcription start site on a base pair resolution. And when we capture these sites and sequence them, we got many tags. Once we map them, we can see on a single base where the transcription site, and it also gives us not only the qualitative information about where the transcription initiates, but also quantitative information about the expression from each individual site. But how we have a lot of they collected and produced a lot of data, we needed to find a solution how to actually scalably in a large place, in a large set, analyze that. And so we basically, I approached like Boris Lenhart's postdoc Sviatoslav Sidorov here and asked, can you help me like to just to write a small next flow pipeline which would do that. And in the meantime, as it expanded, we ended up like with this pipeline here, which takes multiple aligners, which have like many steps and stuff. So, and at some point, and it ends up actually in KJAR, doing a KJAR analysis. KJAR is the bioconductor package to analyze cage data. It was originally developed by Boris Lenhart's group, but in the meantime, Charles Plessy from Okinawa, who is now currently in Okinawa, to take care of developing it further. However, we are familiar with that. I'm also helping maintaining the package. So we are familiar with the ways of processing. So we wanted to include it in the pipeline so that we actually do all the steps which are included in the KJAR pipeline. So, and at some point, I was contacted by Katalin Ferenc from Anthony Materdier group from the University of Oslo, who is also exploring some of her own cage data, and she asked if she could join us, help further developing the pipeline. And so Slava and Catalin are currently like mostly working on developing the pipeline. Slava is working on the next flow part, and the Kata is working on the KJAR part, and I am like supervising and overseeing the entire procedure. And so we are currently like in the finishing phase of developing. And basically the entire pipeline was actually done as on an NF Core template. So at some point, I wanted to ask Matthias actually to have a look what he thinks about it, and if it's actually how the pipeline looks in terms of NF Core standards. So with this, we actually have our different wish list of species, of course, to collect the cage data, sequence or produce the data. Of course, we didn't manage to get all of this wish list species, but we managed to produce quite some few new species. So the green marked ones are the ones which we collected and produced from scratch, while the orange ones are the ones which are already available either from Pumptom or from funks. And so we can now proceed to our evolutionary explanation. So what we are actually doing, what biological questions we want to ask. So it's how the promoters are used in embryonic development, tissue and with the Damir recode in tissue regeneration, but also, for example, in animal domestication, because we were involved in aquafunk, so we have many aquaculture fishes. We are also producing some experiments to see like the regulatory response of zebrafish on toxin exposure, and also look at some cancer data to look on promoter architecture. And what we figured out that as far as we go down, that if we look at the invertebrates, for example, Drosophila or other invertebrates, their promoters look totally different, but with some common features. But we came along with some protozoan samples as well. So we also decided that we will characterize protozoan promoters, and so far we did cage on Crolimax fragrantissima, which is a close relative to all the animals, and we found actually that some features like TCT initiation, which is characterized for translation machinery, or enhancer RNAs are also present in protozoans before the emergence of multicellularity. So this is also one aspect of what we are looking for, and we are also like doing some more, let's say, exotic species like sea anemone or like Dictyostelium discoideum, and so that so far in evolution how we are going currently, and we want to explore it using classical approaches, but also nowadays using AI and AI approaches and the foundational model. I didn't want to go into too deep because I don't want you to listen to me for too long. And yeah, this is the currently where we are. And with that, I would just like to acknowledge my my group, especially Marta, who made my PhD student Marta, who made the cage dataset for the adult tissue in zebrafish, but also my postdoc Katarina and Ira and Alba as well, and my entire division at the Ruđer Bošković Institute in Zagreb, but also other collaborators. And with that, I'm open for your questions.
Damir, thanks a lot. This was a great talk, actually. This was very, very dense, and I could spot many bits and pieces that could be an entire talk of their own. So thanks a lot for showcasing the collaborative aspect of NF Core, allowing people to keep collaborating on the same pipeline a long time across sites. That's a great illustration. Now, do we have any questions or comments? Matthias, I saw that you had a comment on the future of the pipeline. Do you want to comment further? You posted something on the chat.
Yes. So, hi, Damir. My idea would be that we just merge over all the changes, check that it follows the template, because it's based on a fork, right? It was forked from the NF Core CageSec pipeline, and make that then NF Core CageSec 2.0.
Okay.
I mean, it's a big issue, these forks. And so, for instance, we had this happening not so long ago. Jose, I want to comment on this. So some colleagues from Australia Biocommons forked our protein structure pipeline. And so we talked with them and we agreed that it will be a nice thing to put all of these things together. That was relatively easy because the function that they added were somehow added on the top of what we had. So there was absolutely no problem putting everything together. But we have another pipeline. We have the differential expression pipeline that we developed with Jonathan Manning at Sequeira. And this one, we've been working for almost a year trying to bring together what we want to add to the pipeline and what is already in the functional pipeline. And that's a case where a couple of times we have wondered, is it worth having a new pipeline or is it worth making the effort of merging? In general, it's always worth making the effort of merging, of unforking. But of course, it's a little bit more work for a while.
Yeah, so this one would basically be more of a rebase, if not even more, because yeah, we would completely replace the old code because it's still DSL1. So that would, I think, not be too difficult to figure out what will be kept, because it's, yeah, basically nothing.
Yes, except that you have to make sure that it does exactly what it used to do, which is not as trivial as it may seem. This is something we've been fighting with a little bit. But yes, that's the way to go. Does anybody have any other question or comment?
I have a question.
Yes, who's talking? Who's talking?
Praveen. It's Praveen here.
Praveen, please go ahead.
Why you choose Chrome HMM for function annotation, and have you tried any other tools, for example, like deep learning based tools or something like that? And have you also validated? Yeah.
Well, when we did that, there were no deep learning tools, unfortunately, yet. So yeah, we tried with several others. Like at that time when we just constructed that, like there was how it's called from Michael Hoffman, Segway. Yeah, it was also popular, but it didn't give us like quite some results as this one. So we decided like just to stick like at that time Chrome HMM was the most popular as well. However, with all these drawbacks like and stuff, we decided like to go with the two combination, like just to do the functional annotation, but like keep the attack 6 as the regulatory region. I mean, nowadays, yeah, I would certainly like to see what's available and compare it with deep learning and maybe even AI tools. Like but to be fair, I didn't really see too many of the. I mean, probably. Yeah, Informer does some kind of prediction, but not really in this sense that it segments the genome. They more focus on predicting the signals the signals and the raw data out of the genomic sequence than actually doing the segmentation, functional annotation, I would say. And I see a lot of people still using Chrome HMM like in the end. So which is like funny in my opinion.
Yeah.
Thank you.
Praveen?
Yeah, thank you.
Yeah, so it's a great question and a great answer. I mean, I had the same impression, you know, when you were showing your data. Would you feel like throwing all of that stuff in some kind of LLM? And I think the answer you gave is quite interesting because yes, these things are not so great at segmenting and all these kind of things. But bouncing on this, one aspect of deep learning I find increasingly attractive and exciting is dealing with embeddings because embeddings kind of allows us to do our usual biology in a different space. Rather than comparing sequences, we are comparing embedded objects in some multi-dimensional space, and the embeddings can be extracted from any LLM you can train, you know, LLM that have been trained by others like Informer or some parts of Informer, LLM that you will train yourself. Are you tempted to throw all of this data in some kind of LLM training strategy?
Yeah, we are. I mean, for example, since I'm mostly interested in promoters, like there is at least 3 deep learning models, like not really, actually one is, I think it's like transformer based, but the two others which I know, like one was published in Science.
Evo 1 and 2, no? They are typically the kind of things you can really extract a lot of embedding data in these things, and you probably need deep learning to use your embedding data.
Yeah, so regarding the large foundational, like non-specific models, I am actually playing a little bit currently with nucleotide transformers. But yeah, want to test like Evo 2. Didn't have the chance yet to test it because we had a journal club a couple of days ago here, and like, yeah, okay, it somehow has an answer to everything, but when you look at the position, like FastCon is still better than it, you know, like in predicting functionality. So
yeah, we looked at it for gene prediction, and it is surprisingly good, but surprisingly not that good as well. You know, like
exactly,
that's
yeah.
the thing. But that's kind of the problem with deep learning, with LLM in general. No, they are almost dangerously good for not being good enough. Like 99% is great, but it's actually much more dangerous than 80%.
What I kind of want to explore, like if I can refine some findings I get with the classical methods. So if it could give me, let's say, if I put it as a semi-supervised method, like I provisionally like assign classes with the classical method, if it can actually improve those annotations further based on the sequence, the motif composition, and to learns. And basically, if I do that, of course, I want to know based on what it learned it. So without actually a good explanation tools, like I would be very reluctant to use it as a black box. So that's all.
Yes. Do we have any other question for Damir? Unmute yourself if you have a question. And I had one more in the meantime. So I was stricken by the symmetry on on the maps you were showing, on the UMAP maps you were showing. And so you explained that this had to do with the model not distinguishing 5', 3', and 3', 5'. But the symmetry is nonetheless remarkable. And it's remarkable because it's almost perfect, but not perfect. which suggests some kind of constraint, no?
Yeah, yes and no. Like I would expect actually if I plotted the PCA or something more linear that actually in the PCA the symmetry was much more prominent. I would assign that to like this.
So that's more like a UMAP distortion.
Yeah, it's more of the UMAP thing, I would say. Than
you should send it to your pastor because he keeps complaining. It's a good actually. It's quite a striking example that of UMAP distorting data. Very interesting.
So we basically, I mean, I try to use it for anything else, but only visualization. But in the end, we just decided it's a nice way to visualize the data. I mean, if you look at the density and do some kind of density clustering and stuff, it might give some useful information and some. I wouldn't say representations necessarily because I wouldn't trust like really to do some calculation on that, but just like based on the density on the plot, like to try to, I mean, which kind of again is like kind of using a representation for prediction, but like some neighbor-based clustering method to try to predict some, let's say, hubs in the in some regulatory space, let's say, to distinguish maybe locally the different classes of enhancer or promoters. And it shows some developmental time course because it has the gradient of activity of like less from less active to more active, but yeah, then it tries to distort that based on the symmetry and everything else. So
yeah. Anyway, that's quite interesting. So I have, I don't think we have any more questions. I have a last one, but that's really out of curiosity. My understanding has always been, but I'm really not a specialist, so I quote this more in pub talk and stuff, that Dario Moreno can go haploid, no, if exposed to some X-ray or some kind of stress. Is this correct?
yeah, I think so. But to be sure, I won't. I don't want to give like I haven't analyzed any of this kind of.
Because that's quite a remarkable property, a capacity, and I was just wondering if it has an impact on the organization of the genome or on its regulation. The fact that it has.
Not actually sure because like most of the experiments data set and everything like which I was exploring were either some mutants where they needed to have like F1 generation mating with the wild type, so they needed to be deployed. And yeah, we are also looking a lot into maternal to zygotic transition, where again it's important that you can separate like the maternal from paternal stuff. So like not sure about the haploid. I know that there are some ways, but to be fair, I am not really familiar with this line of.
Okay, no worries. Thank you. Thanks a lot, Damir. This was a great talk and very insightful for our community. I mean, it's a talk that really showcased the connection between the generation of high quality data and the connection of this high quality data with the data globally produced in biology that requires very, very high levels of interoperability so that data sets can be compared. And as Dobzhansky used to say, you know, nothing makes sense except in the light of evolution. And evolution is comparison, and comparison is data interoperability. And I think you made a splendid showcase for this to occur if fish data is produced the right way using NF core tools. Other tools, of course, it doesn't matter, but as long as things are analyzed in a way that makes data interoperable. So thanks a lot for this great talk. I'm very happy to announce that everybody stayed during the entire talk. We didn't have the highest attendance we have because it's the middle of Easter holidays, but we had 15 people today. And I was looking at the NF Core Animal Genomics group that now has 113 members. So this is growing quite a lot. Next month we'll be hosting James Ricci from Iowa State University. And so it will be a pleasure to have him. James is Associate Vice President for Research in the Department of Animal Science, and is a very, very well-known specialist of animal genomics. And I'm sure this will be a very enjoyable talk. Jose, is there anything you want to add before we close this?
No, I think yeah, everything is okay.
So the talk will be made available on YouTube, and of course, if you have any question or anything, just let us know. Also, as always, we should make this announcement. If any of you have something interesting to say along the line of the talks we have heard today, connecting data analysis and tools, please reach out to us. And we are quite full till next year, but starting from next November, I think we are beginning to have slots. So we have to find interesting talks for these slots. And at the end of the day, we are this channel. So please reach out to us if you have anything you want to share with the community. Thank you all. Have a great Easter if you're celebrating it, and see you all next month. Bye-bye.
Bye-bye.
Bye.